Hello, everyone. Good morning, good afternoon, and good evening. My name is David Good. I am a Senior Director of Business Development here at Aliri Bioanalysis, and it's my pleasure to serve as your moderator today. We're really excited to have you join us for what we hope to be a great discussion centered around whole body pharmacokinetics. And for anyone who's just learning about Aliri, we are a premier global CRO that offers comprehensive bioanalytical, spatial omics, and biomarker solutions to enable and enhance strategic decision-making, strengthen regulatory submissions, and accelerate drug development for our customers. We are proud of our scientific strength and achievements over the years. We actually just celebrated 30 years of operation last year. You may recognize us as one of our three legacy brands that you see here on the slide: Pixent Labs, EMA Biotech, and Tandem Labs. So in this session, we're going to cover how mass spectrometry imaging of whole body mice offers an effective experimental setting for comprehensive characterization of drug pharmacokinetics at an early stage, which facilitates better and faster drug candidate selection. This webinar is being recorded, and we will share the recording for your future reference after today's call. I know that was one of the questions that was in the Q&A. So please, mentioning that Q&A box, we do have a Q&A box that will be open throughout the webinar. So feel free to pop in any questions you may have for our presenters into that, and we will attempt to address everything at the end of the webinar. We do have two great speakers for you today. Mathieu Gaudin, Ph.D., is Laboratory Director for Aliri site in Lille, France. Mathieu has more than 14 years of spatial bioanalysis experience. He leads lab operations at Aliri Center of Excellence in Imaging in Lille. And Mathieu is an expert in mass spectrometry and a subject matter expert in spatial bioanalysis, DMPK, metabolomics, and lipidomics. We will also be hearing from one of Aliri's customers, Hervé Giorgi Ph.D. He is the Director of Non-Clinical Development at Genfit. Hervé has spent more than 30 years in drug development and manages Genfit's technology platform, including chemistry, analytical chemistry, and bioanalysis. And he is ultimately responsible for Genfit's safety activities, including toxicology and safety pharmacology. So with that, it's my pleasure to give the floor to Mathieu.
Thank you, David. I wanted to start by some numbers. 83%, that's average attrition rate that was considered in this paper reviewing the pipeline of 4 major pharma companies at the preclinical stage. That's really an impressive number. We know it can be even worse. And we know that as drug developers, we should do better. We should do better for moving drugs towards the patients. And one interesting finding of this review was the drop of consideration of PK parameters from preclinical to clinical phase. On one slide, you see that at the preclinical stage, PK is not really the major factor of discarding drugs from the pipeline. And however, in first-in-human, it comes back to the third place as a reason for stopping the drug from progressing any further. So what happened in the meantime? Were the tools, were the models, were the techniques not good enough in preclinical stage to predict the failure that was going to happen due to PK reasons in phase 1? That's a question we were asking today. And by presenting you one tool, I hope you will have the opportunity to have this additional opportunity in your toolbox. It's not a miracle tool, but it's definitely a very useful one. I will try to show you with 5 different use cases how mass spectrometry imaging applied to a whole body experimental design will help you, hopefully, to make better and faster decision-making to de-risk your program. So to start with, I will remind to those of you who are not familiar with mass spectrometry imaging how it works. So today we will be focusing on the specific case of whole body. So we start by a whole body mouse that we will section, mount on a microscopic slide, covered by an automatic sprayer with a matrix, and the sample is introduced in a mass spectrometer, not any type of mass spectrometer, MALDI, FTICR. That's the two significantly big instruments that you see on the bottom right picture, two Solarix instruments from Bruker. We are lucky enough to be the only CRO in the world to be equipped with two instruments like this. And they have a unique capability of providing high spectral resolution data on every single position of the tissue section. So we collect literally a molecular fingerprint associated to XY coordinates in every position of the tissue. Once we obtain that, we move to the data analysis part. Next, please. Yeah, we combined all the different information acquired locally into an average spectrum, and we can then start interrogating this very rich data set. And every single peak that you see on the spectrum will lead to a distribution map that you can see on the right, a couple of examples. And the main advantage is the specificity. We see multiple molecules, thousands of signals are detected. And one take-home message from this presentation is really that the technique is label-free. We all know quantitative whole-body autoradiography is a gold standard in the industry for tissue distribution at late stage. But it requires radiolabeling. It requires carbon-14 or tritiated molecule. Here we are talking about a molecule that use cold compound that doesn't imply any additional synthesis cost for drug developers to be implemented in their drug development pipeline strategy. So it's a non-label technique which adds molecular specificity. And we will see in the next example the benefit of this. Why is it interesting to see so many molecules? Let's focus on the drug-related molecules. Here you see on the first column, the drug distribution image. But at the same time as we image the drug distribution over time, we can simultaneously image the drug-related metabolites. And why doing so? It's because they do not have the same behavior. Sometimes on the 2-hour time point, for instance, after the distribution, which is more or less similar until 1 hour, we start seeing differences in distribution between the drug and the metabolites. And specifically, the drug crosses the blood-brain barrier. We have a very significant exposure of the brain, which is one of the bright region on the right of the image. Whereas the drug metabolite is absolutely not crossing the BBB. So we start seeing differences in tissue pharmacokinetics by using multi-mass spectrometry imaging and having the specificity provided by mass spectrometry, contrary to QWBA, which would have given you the sum of the two images that you see here. So these images show you the absorption, the distribution, the metabolism, and at the late stage, you see the elimination phase with the compounds and the metabolites being distributed in the kidney and in the intestines, suggesting the elimination routes of the drug candidate. So the image already tells you a lot, but you can extract numerical data from these nice images in order to support the decision-making. You can have, on top of the traditional concentration time profile on plasma that you have in red, you will be able to plot all the concentration time profiles in every organ. And what we observe routinely is that clearance, half-lives, all the PK parameters are very different from one organ to another. And it leads us to partition coefficients for every organ, and that's a great opportunity to give this data to our PBPK modelers so that they can refine their PBPK models and validate their predictions of tissue exposure. And in order to ultimately have a more accurate first-in-human dose selection. It can also support tox investigations, and one of the examples will show you how. So multiple case studies will be presented, 5 of them, that will illustrate 2 major categories of use case. Problem solving. As a CRO, we are contacted by sponsors that have a question. They may have difficulty to interpret their existing data, and we are producing additional data that will help interpreting in a more global way the behavior of the compound. An interesting or the use of the technique is in a more proactive manner. It's to de-risk the drug development program by anticipating possible issues. And I will illustrate that by showing you a case in terms of efficacy in order to confirm the target organ exposure, but also safety to anticipate the possible accumulation of drug-related material. And a last case will be more oriented toward formulation and dose selection. An advantage also of the technique and why using mass spectrometry imaging instead of the classical other approaches is its flexibility in terms of timelines. We already mentioned that it's a label-free technique that requires no radio labeling. But as a consequence here in terms of timelines, you see that it's much faster. The method development plus the sample analysis takes 2 to 3 months, depending on the number of animals to be imaged, which is in any case much faster than using a mass balance study, which is heavily limited in terms of timelines by the time requested for doing the radio labeling of the compound, which takes time and which is also very difficult to forecast. It can take few months or it can take half a year, depending on the difficulty of the synthesis. So it's really difficult to make predictions on when you will get your data. With whole-body PK, it's much easier for you to know in advance when you will have your data ready because its technique is much more predictable. Now we will start with a different case study. So QWBA, as I said, is really the gold standard in the industry for drug distribution. So definitely we do not compete with this technique. We want to work as a complementary technique. Why so? Because the radioactivity is a great tool for measuring quantities, but it doesn't bring any molecular information with it. If the radio label is retained, the signal can come either from the drug or the drug-related metabolites, and you have no way to know it. So one possibility of smart combination of the 2 techniques is to use mass spec imaging to provide the molecular insight that QWBA was missing. And in this example, we have been treating rats with tritiated chloroquine. And we have a nice colored, artificially colored image of the radioactivity detection of chloroquine. And when we do QMSI, so quantitative mass spec imaging, on top of it, we see that it can be decomposed into 2 components, the drug distribution and the metabolite distribution. And if we take spleen as an example, so it's as a round-shaped organ that we have here, we see some radioactivity, not much, but still some. But when interrogating the other data sets, we see that only drug is present and the metabolite is not distributing in this organ. That's one example. Kidney is more exposed to the drugs than it is exposed to the metabolite. And we see liver, for instance, as a combined exposure of drug and metabolite. So mass spec imaging will really bring you an additional level of molecular understanding that is dramatically missing to a QWBA. That being said, that example is pretty a niche one. One much more common example that I would like to present is understanding limited bioavailability that you can observe on your compounds during your drug development pipeline. So as you know, oral bioavailability is a combination of absorption, fraction escaping the gut metabolism, and fraction escaping the hepatic metabolism. But when with your plasma measurements, you measure IV route, oral dosing, you make the ratio of AUCs, you obtain a low bioavailability, you still do not know who is the culprit. Is it the low absorption? Is it the heavy metabolism at an early stage in the gut? Is it first pass metabolism in the liver? When it's important to know in order to orientate the pharmaceutical development strategy. Do we have to reformulate the compound to ameliorate absorption, or is it a metabolic stability the issue? And is it then a medicinal chemistry topic to solve it? Here, we can help decision-making by this very simple design, having 6 animals orally dosed, 6 animals IV treated. We collect the whole body mouse carcasses that would be subjected to quantitative mass spec imaging. We collect at the same time the plasma that undergoes classical bioanalysis by LCM-SMS. And what have we seen? We have seen definitely that the compound was restricted to the gastrointestinal tract and the stomach. The images were extremely bright in these regions, but with very limited exposure of the other organs. On top of that, taking advantage of the molecular specificity of mass spec imaging, we investigated the phase 1 metabolites, and none of them were detected in any tissues. So it was a very, very good evidence that limited oral bioavailability was really due to limited absorption and not an issue of metabolic stability. And all of this information were obtained in one single experiment instead of multiplying mate-ID on one hand, in vitro absorption on the other. So that was really a comprehensive overview in vivo of what is going on in the tissue and without having to wait for the mass balance data. That was the 2 first examples were in the problem-solving category. I would like also to talk about efficacy, which is more positive. We want to make sure that a sufficiently high amount of drug is reaching the site of action, which is a critical factor to expect some efficacy, of course. And by implementing in this oncology program a tumor on the flank of the animal, in the very same animal experiment, we are able to obtain a section through the tumor and get drug concentration measurements at the site of action. And in the classical sagittal plane of the animal, all the other organs. So we make the best use of the same animal experiment, reducing variability, reducing ethical issues by having efficacy and PK data obtained on tissue with the same animal. And we have been able to confirm that the concentration was high enough to expect efficacy and stop tumor growth, and characterize at the same time the PK in the different tissues. I also mentioned that during my first slide on attrition, the first cause of termination of a compound was toxicology or clinical safety concerns. So that's important to inform these very critical aspects using all possible tools available. And this whole-body pharmacokinetics tool using mass spec imaging can also be extremely informative for anticipating tox issues. This is one of the best examples we have. Classical design with several time points, 1 animal per time point, oral dosing. We see a wide distribution of the compounds in multiple organs. Nothing really surprising, until we zoom in a little bit into an unexpected finding that we see a surprisingly high accumulation of compound in the ovaries. And the flexibility of the technique is illustrated by the ability to take an adjacent section to the one used to obtain the classical distribution and zoom in, do a high spatial resolution image, correlate it with hematoxylin and eosin staining, and observe a nice correlation between the white hotspots that show the highest levels of drug, perfectly correlating with the histological structure of attributed to ovary follicles. So it was not only an accumulation in the ovaries, but an accumulation in the ovary follicles. I'm not a toxicologist. I'm an analytical chemist by training, so it's not my role to say that it's a tox finding, but for sure, it's my role to inform my sponsors that there's possibly something very intriguing and that it should be considered when designing the tox package to make sure that the protocol will have a special emphasis on the reprotoxicity in order to investigate if this drug accumulation presents or not a safety concern, but at least it increases the chance of not missing something extremely important at the tox stage, and if necessary, stop the compound at the appropriate moment. So it illustrates the flexibility of the technique, starting from a very holistic approach, very classical PK question, and then with agility, making the design and the question evolving toward a more data-driven approach in order to answer the toxicity question that was rising during the project. Last case study I'd like to show you, it's not tox, it's not efficacy, it's a more question of formulation. We have some sponsors that are developing technology platforms, absolutely amazing, with very specific delivery capabilities. And they want to document the added value of their platforms over classical formulations. And here, by comparing a group of 6 mouse treated by the native formulation and 6 with their innovative formulations, we have obtained with a time course the exposure of the different organs, and we have been able to document the fact that the target organ, which was a tumor in that case, were more exposed to the drug when at equivalent dosing, when using the liposomal form instead of compared to the native formulation. And that type of comparison has been done on multiple organs at the same time. So it provides scientifically robust data to support claims in order to document better targeting, better exposure, better kinetics, lack of accumulation, whatever you can imagine, for a new innovative formulation. It works exactly the same way for documenting the added value of prodrugs or any other delivery systems. So now I hand over the presentation to our customer, Hervé, with whom we have been working, and who will present his personal view on the added value of mass spec imaging for him in his drug development strategy. Hervé, the floor is yours.
Yeah, thank you, Mathieu. Yeah, so a lot of things already discussed, so I will just give you our feedback and why we use MSI in our strategy of development. First point, we use it at a very early stage when the first formulation for in vivo use are available. Of course, for the toxicologist, you know that the in vitro genotox package is key and most of the time go-no-go decision. So here in Genfit, we will perform first a genotox package, and then before the general tox package for the first in human, we implement this technology. And why? The first reason, as it was explained before, is that we are able to compare different routes of administration. First, the intergene route, of course, and some alternative, and to see and compare the differences in PK and tissue distribution. That enables you to select the route of administration that seems the most appropriate for your indication, and to see some potential effects on pharmacology, see the efficacy, and have a safety concern. So I will give you some example. Yeah, regarding efficacy, it is very interesting to see the tissue distribution, and of course, if it is in line with the intended therapeutic use, and particularly, does the drug meet the target? So it's not only a question of systemic exposure, but it is always nice to ensure that your drug goes at the right place. And in addition, because as explained before, you look at the tissue distribution over time, over 24 hours, you can see how long is the organ or tissue of interest exposed. And so that it is also interesting to adapt the dose or the dosing regimen to improve the efficacy. And in addition, on top of that, you can anticipate potential off-target and target organs in view of the tissue distribution. And so that you can anticipate these potential tox effects in your future general tox package. You can also see if there is a risk of accumulation, depending on the elimination of the compound, and particularly if there are some tissue of organ you need to pay attention to. And so to just summarize, all the useful information we get from this study is really to balance the efficacy and the safety. It helps you to define or refine the route of administration, but also the dosing regimen to balance safety and efficacy. It can help you, as described before, to select some formulation and to anticipate risk of toxicity, and particularly to know in advance if there are potential target organs, and also give you for efficacy because you can see that your drug distributes to the right place. In addition, no need to get at this time a radiolabel compound so that it is quicker than a mass balance or autoradiography study, and of course also less costly, and it's really simple to organize and manage. Just one point of attention, in case you want to explore the IV route. Sometimes compounds are highly bound to proteins, and in that case, I would recommend you to perform a protein binding assay before performing the MSI, so that you ensure that it is appropriate. Otherwise, you may have a loss of sensitivity or difficulties to detect your compound in mass within the whole body.
Thank you, Hervé. My conclusions will be very similar. Really key take-home message, it's a label-free technique. So it gives you a considerable flexibility to apply it whenever it's appropriate for you in your drug development strategy. Of course, it can vary from one company to another, but I fully agree with Hervé, before the tox package is really when it is the most appropriate to me. Being label-free, it's very cost-effective. It's fast. We have compared the timelines compared to a classical mass balance, considering the fact that radio labeling is really a limiting factor. And also the design is very flexible. We have been able to switch from a PK question to a TK question in the middle of a project based on the data we have observed. So it's really something to consider in terms of good value for money. The scientific wealth of information provided is really, really important. As a summary, the different case studies we have seen, so supporting QWBA data, but more interestingly, at an early stage, understanding the limited bioavailability, working on efficacy with target organ exposure, working on the safety part with understanding the possible accumulation in specific organs, and more generally supporting not only the decision-making in drug formulation, but any decision-making related to pharmacokinetics, which, as we have seen in the attrition data, is something that is maybe underestimated at the preclinical stage and becomes absolutely critical when moving to first in human. So thank you for your attention, and we will be moving to the Q&A session.
Thank you, Mathieu and Hervé, for a really engaging, informative session. Really, really interesting. So we do have quite a number of questions that have been posted in the Q&A. And I think reading through these, I think some of them maybe Mathieu and Hervé, we can address together. So one of the questions is, can all drugs be used in this technique? How can you determine what drugs are good candidates for this technique? But hold on, Mathieu, because there's another question that's related, which is Aliri MSI limited to small molecules? Can it be applied to biologics? So perhaps you could tie those two together in your response.
Sure, with pleasure. So yes, I would say that it's limited to small molecules. If you browse the literature, you would find some niche examples of protein imaging. But as a CRO, I would not encourage you to go that path, because it mainly works only on highly abundant proteins. But we are all interested in biologics, very efficient, very effective, and administered at low concentration. It's pretty unlikely to work in most cases. So I would really encourage pharma to focus their efforts on small molecules for application of mass spec imaging distribution studies.
So Mathieu, just a leading question here, however, small molecules does not necessarily mean NCEs. We can or cannot do this with peptides. For instance, what is the limit for peptides in terms of molecular weight?
I'm comfortable working up to 2,000 Daltons without any issues. If it is locally applied, we can probably give it a try even higher. But yeah, it's really something we are open absolutely to test. That's all purpose of the method development we apply for all our studies.
All right, fantastic. So another question that I think actually, many of our attendees will find interesting or informative is quantitation, right? So how quantitative is this technique? Could you go into that a little bit for us?
Yeah, when the company has been founded 15 years ago, sorry, it has been founded on the belief that the technique would be useless for pharma if we were not able to make it quantitative. The belief was that a nice image is never good enough for a pharma sponsor to be used in terms of strategic decision making. We need to provide figures. So that's why all the efforts we have done in the past were fruitful. And now we can definitely say that the technique is quantitative. We have demonstrated that multiple times with cross-validation compared to LC-MS, compared to QWBA. So now the community accepts that the technique is quantitative, which was not the case a decade ago, but now it is. And we have patented several approaches to make it work. So yes, it is quantitative, and that's what allows us to determine AUCs expressed in a microgram per gram hour, and do some PK parameters determination based on absolute quantification.
Fantastic, thank you. So I've got another few questions here that I think are linked. Okay, Mathieu. So you and Hervé discussed this really within the context of mice. However, I have several questions here that are, is this only available in mice, or are rats an option? But hold on, because really, if you could answer that in terms of how do we do QMSI for other species, right? And what approaches do we take for doing this type of work in different species other than the mice that you presented?
So we can do it in rat. It's only a question of size, obviously. It's twice as big. So we have to adapt the sectioning strategy, but it is feasible. So we have a little bit more work in sectioning, a little bit more work in data analysis to reconstruct the entire rat. But it's an option if it is mandatory. It's more cost effective to do it in mice. But if there is a scientific rationale to do it in rat, we can follow you in that direction for sure.
And then for larger animals, I guess this is a leading question for you, Mathieu, but for larger animals, obviously it would not be whole body. However, we can look at tissue or organ sections of interest or even resected tumors and do this work, correct?
Yes, it as to be totally transparent, the largest size I can accommodate in my instrument is the size of a standard 96 well plate. So it gives you an idea of what we can enter into the instrument, physically speaking. So I've already analyzed a pig kidney, sheep brain, non-human primates lung. So any isolated organ from large species can enter the instrument for sure, not whole body, obviously.
Fantastic. Thank you for that extra clarification. So then, we have a couple of maybe more technical questions as well for you. One being, how do we deal with matrix effects using this technique?
That's a very good question because intrinsically, when you look at an image on a whole body mouse, you have multiple organs. That's the entire purpose of this study. So you have different matrix effects from one organ to the other. So if you do not have a strong normalization strategy, there's not a way to produce reliable data. So we have not reinvented the wheel. Mass spec imaging works with isotope label standards. The know-how is just to use what any bioanalyst with LC-MS/MS would have done with a carbon-13 or a deuterium labeled compound and apply it in the appropriate way on the tissue and use it for normalization in a normalization strategy, point by point, pixel by pixel, to compensate for matrix effects in every organ.
Fantastic, thank you. Another topic which I'm seeing several times within the Q&A box is really related to sensitivity, LOQ, LOD, right? And so I do know, right, that this varies depending on the compound type, right? And so maybe you could give a little bit of framing around that in terms of whether these are small molecules or the peptides that we're discussing or something in between.
It's totally compound dependent, as you said. No surprise. It's mass spec based, so it can go anywhere between 20 nanogram per gram up to 1 microgram per gram. That's a pretty typical range. So we are in the hundreds of nanogram per gram in for most compounds. Sometimes, unfortunately, we have compounds that are not really appropriate for mass spec imaging, and it's a challenge. So we have several tools in our toolbox to enhance the sensitivity. Derivatization is one option. We have several others, but yeah, we are playing in this hundreds of nanograms, tens to hundreds of nanograms range for the most appropriate compounds. For peptides, we are more in the tens of microgram per gram, but it's just also a question of molecular weight.
Okay, thank you. Moving on here, another question that is being touched upon by several people is the resolution, right? So what is the standard resolution for this technique, and could you compare that to QWBA, for instance?
The instruments are flexible. That's really something that I will say several times. So because we can go down to 20 micrometer, but it will take forever if we image the entire mouse at this spatial resolution. So we have a more screening and efficient approach, running at a few hundreds of micrometers of spatial resolution. So it's typically 350 for the mouse. And then, as we have illustrated in one of the case studies, if we see something atypical and we want to focus on it, we can go down to 50 or 40 micron very easily and refine the question. But it's a balance between the size of the sample and the spatial resolution we want to use to address a specific question. It's really a combination of the three factors. We don't run a full image at very high spatial resolution just for the sake of producing a nice image, because it's not cost effective and not scientifically sound.
So Mathieu, just as a in addition to that, maybe do we have the ability, for instance, right, to distinguish between interstitial space and organs, and perhaps different suborgan types, right? So like within the brain, the hypothalamus versus something else. And are we able to, for instance, there's a specific question in here about being able to distinguish whether or not a molecule has actually passed the BBB or whether it's accumulating at the BBB, right? Looking at something and seeing whether or not it's in a vessel or it has actually left the vessel itself into an organ of interest.
Very good question. We will use for sure mass spectrometry imaging, but not in a whole body setting. If we have a CNS-oriented question, we would prefer to work on the isolated brain, flush the brain in order to evacuate all the remaining blood from the vessels and have a group flushed versus non-flushed, and do the sagittal or coronal sectioning, and really have the ability to focus on histological regions with more flexibility using an isolated organ. But definitely mass spec imaging is a good tool for investigating BBB crossing, but the whole body animal doesn't give all the flexibility to select the right histological regions in the brain. So I would do it differently on the animal part of the study, but for sure, mass spectrometry imaging is a good tool to answer that question.
So this question actually may be for Hervé, which is, you know, Hervé, you mentioned protein binding towards the end of your discussion. What is your observation in terms of what is the limit, right, in terms of percent bound that kind of disqualifies this being a reasonable technique to use in your study?
In fact, it depends on the rate of protein binding. If you face a compound that is mainly, I would say, 99% bound, and it was the case in the past, we had some highly bound compound. For sure, it is a challenge for MSI by the IV route. Oral is different, but so it depends. You have to know, if you know before that your compound is just partly bound, so you can discuss with Mathieu and see whether you can reach the threshold, so that you can be above the limit of quantification. It will depend, and you will start by a first step with method validation, so that you can ensure before you start the whole body that your compound is adapted. Mathieu, you can confirm that you will start by this assay before engaging a full MSI with a potential bound compound.
To complement this answer, the very, very minimum requirement is to be able to provide me with 5 milligrams of compound. That's the bare minimum I can't work without. If you can provide us with an LCMS method, if you can provide us with some protein binding information from some meta-ID information, that's a big plus. But meta-ID is not requisite as a preliminary step. If you have this information, I will be able to exploit it and provide you more metabolite information in return. So you extract more value from the study, but it's not necessary to have this information before. Same for protein binding. It really informs what is going wrong, what is going right, how you interpret the data. If you have it before, you can use it as a decision-making process. But yeah, the very minimum is just to have the powder to give us.
Fantastic. So I think we've got just a couple of more that we have time for, but kind of feeding off of your bit there about MetID, Mathieu, we do have a question asking whether or not this technique can be used to mine for metabolites or whether we need to know them a priori.
I would say that we do not believe this technique is going to replace QWBA. Same way, we do not believe that this technique will replace classical MetID by LC-HRMS. Doing it with the chromatographic dimension offers much more guarantees in terms of isomers identification. We do not have any chromatographic separation. So if I look at metabolites, I will have less molecular resolution than a classical MetID experiments would do. So using this tool to determine without any prior knowledge metabolites is not the most appropriate. I can do some work with it for sure, but it's not a standard approach and it's not the most cost effective. But it's useful to have a discussion to see if we can combine smartly the two needs in one. But we do not aim at replacing the standard MetID approaches for sure.
Thank you. And so I think this will be our last question here, which is a number of people asking about molecules that may have, for instance, low ionization efficiency, right? So how do we do that? And specifically, for instance, what is our experience with utilizing a chemical derivatization step, right, to enhance the signal for certain molecules of interest?
I've been playing with chemical derivatization for 10 years, so I like particularly this topic as a chemist for sure. So yeah, that's one of the options to enhance ionization. It has to be carefully applied because yeah, it comes with additional level of complexity. We are just complexifying the tissue, but at the expense of this complexity, we obtain more ionization of some families of compounds that react with our derivatization agent. So it's definitely one of the ways to move forward when we have lower ionization, provided we have reactive groups on the molecule, which is not always the case. But and some molecules just cannot be seen without derivatization, at least in standard MALDI. Now we have some other post-ionization techniques that could offer other options, but yeah, it's really one of the major tools in our toolbox for method development.
Fantastic. Thank you so much. So with that, I would really love to thank everyone for really engaging discussion and number of questions today. We really look forward to seeing you at our upcoming webinars, and thank you for attending this one.